Cool. Let's get into early brain development. So these are the things that we're going to be covering for early brain development. So this is separate than the neuronal circuits. That's going to be a whole different list of objectives. But we're going to talk about stem cells for a bit. Pluripotent, totipotent, induced pluripotent stem cells. We're going to talk about all those different types. Well, we're going to focus on the ones that are most important, like pluripotent stem cells and induced pluripotent stem cells. Then we're going to talk about transcription factors and signaling, right? Transcription factors are going to be really important for allowing basically a specialization of cells, right? Specialization as well as differentiation, where you go from a progenitor cell and you can differentiate it into like a more specialized type of cell. So we're going to talk about different types of transcription factors that play into that. And then we're going to talk about embryogenesis. So the process of gastrulation, where you have 3 different major layers being formed, the ectoderm, mesoderm, endoderm, and then what those eventually arise into, right? Like the ectoderm turns into the neuroectoderm, which eventually gives rise to the brain and spinal cord. Okay, mesoderm, you see the notochord in there. And then we're not going to really talk about the endoderm because it's not much high-yield stuff in neurology with the endoderm. Then we're going to talk about a little bit of regional specifications in brain. We're going to, again, talk about the genes, right? You know, there's lots and lots of genes that get involved in here, but we're going to kind of briefly talk about a handful of them, the ones that, you know, I want you guys to mostly focus on. Inductive signaling, migration, neurogenesis, and then we're going to talk about some clinical stuff, things that might happen if the genes and the transcription factors don't work properly, right? There's a handful of really nasty conditions like holoprosencephaly, horrible stuff, right? Lissencephaly. You don't really see it, but you'll experience it if you try to engage with the individual as lissencephaly. They have a smooth brain, so they're going to have some serious cognitive deficits. What else? There's a couple other ones too that we'll talk about later on. I can't think of. Neurotube defects. We'll discuss that as well. But cool. Let's talk about stem cells now. The main stem cells that we're going to be focusing on are going to be the pluripotent type of stem cell. And with pluripotent stem cell, that's going to be the step after you start with a totipotent stem cell. And then once that actually turns into a blastocyst, those germline cells within the blastocyst are going to be the ones that can turn into pluripotent stem cells. And then once the pluripotent stem cells are present, those can turn into a bunch of different tissue types. So they can turn into any of these different cell lines. So cell lines that involve the ectoderm, mesoderm, and endoderm. So with the ectoderm, that's what's going to be turning into, for example, the central nervous system, the brain, the spinal cord, right? And then you also have mesoderm, which can turn into all sorts of different cells. Like within the mesoderm, you also have the notochord. But then you also have things like the circulatory system, heart, lungs, all that stuff. Lungs are going to be derived from the endoderm. So that's what pluripotent stem cells can do. They can turn into any of these different cell lineages. Once you basically commit to a specific cell lineage, then that becomes a multipotent stem cell. And then at the very end, you only have unipotent stem cells. Okay, so in other words, pluripotent stem cells are going to be some of the most versatile of all the stem cells available. But, of course, we all are familiar with the bioethics that come into stem cell like research and work that involves these types of cells, because they're going to be derived from embryonic stem cells, right? So then that opens up an entire Pandora's box of controversy and issues that need to be addressed. And so there's ways that we're going to talk about later on that you can actually induce pluripotency into cell lines using different types of tissue factors or growth factors and things like that. And so you can actually induce pluripotency into a stem cell. And so it doesn't have to be derived from embryonic stem cell. It could be, for example, derived from fibroblasts. So if you add a cocktail of different growth factors into fibroblasts, you can basically turn them into a pluripotent cell. And then you can, have them turn into other cell lines depending on how you induce them, right? You can do some different things like retinoic acid, for example, and that can help them differentiate even further into they're more mature type of cell. So anyways, neural stem cells are going to be another derivative of pluripotent stem cells, and those can turn basically to any cell type within the nervous system. I'm talking like oligodendrocytes, Schwann cells, astrocytes, etc., right? Neurons, etc. So here's a little interesting study where they've taken somatic tissues, put them into a petri dish, right? So those are somatic cells. If you take out the nucleus of a somatic cell, it has all the DNA in there, right? It can turn into any cell because in our DNA, we have the capability of just basically turning into any sort of cell type in your body, right? And it, depending on what transcription factors your cells are going to be exposed to, that's what basically dictates the specialization of the cell line. So if you take a somatic cell, it doesn't matter what it is, and then you throw it, for example, into an egg, right? Then you're basically going to have all the necessary tools to be able to create a full organism, right? Complete organism, every single tissue type. So in other words, this would be an example of how the DNA can basically contain the information for the entire organism. And so this is what this experiment basically is demonstrating. So kind of cool. And again, here is where we're getting your pluripotent cells, right? This is why it's so controversial, because it starts with literally fertilization. And, basically whenever you have conception, right? Fertilization is where you have conception. So this is an actual living being at this point, right? And so this is why this becomes extremely controversial. But once you get into the blastocyst form, that's where you can harvest those cells, right? If you didn't really care about the ethics, right? You can harvest those cells, and those are the pluripotent stem cells. And again, turns into any sort of cell line: ectoderm, mesoderm, endoderm, etc. Now, in terms of differentiation, this is where you have the specificity of the cell, right? You have to differentiate the cell line. And there's a bunch of different things that can be added to cells to help differentiate them. So like in my lab, for example, we're working with a certain cell line that's derived from, glioblastoma. And it's called SHSY5Y cell line. And to induce that cell, it's an undifferentiated neuronal type cell. To be able to actually induce it and make it differentiate, we subject it to retinoic acid. And it takes I think about 5 days or so for it to actually start. You can start developing the little neurites, dendritic trees, and things like that. So it's really cool. So that's what we're doing up in my lab right now. So this is differentiation, and we're using retinoic acid for that. But there's other things that you can use. There are transcription factors like this one. NANOG is going to be one. That's what's going on over here. Also, nestin can be another one as well. And then, so there's different types of transcription factors that could be used at the end of the day. Now, stem cells, again, they can differentiate into basically any tissue type. And embryonic stem cells, if they're injected into the central nervous system, that's where you can get neuronal development of any tissue that might be, for example, damaged or missing. There's a lot of research being done now using stem cells to be able to reverse, for example, Alzheimer's disease and reverse other types of neurodegenerative diseases. Barrow Neurological Institute here in Phoenix, they do a lot of that type of research, which is super cool. Very promising, right? Because especially if you're taking the patient's own cells and then inducing pluripotency in them, that's really cool, right? Because now you're using the actual individual cells and being able to use those to help regenerate tissues. Now, there's a whole entire area of medicine. It's called regenerative medicine. And that's basically what they're doing. They're harvesting cell lines from the patient and then being able to use those cells to actually treat different types of disorders. I was listening to this really interesting interview the other day on a regenerative doctor. I think he practices out of Dubai or something, but he's really well known in regenerative medicine circles. And he does a lot of things with PRP, protein-rich plasma. And PRP is basically harvested from, you take the patient's blood, then you put it into a centrifuge, right? The top layer is what, you get plasma from, right? Plasma is mostly going to be made up of water, but you have proteins, immunoglobulins in there. But there's also lots of growth factor and repair, like things to repair damaged tissues. And so a lot of people are using PRP to help regenerate like joints, if you have degenerated joints and things like that. So you can actually inject those cell lines into a patient, and it can basically help regenerate different types of tissues that were damaged. Same thing with stem cells. You can induce pluripotency in stem cells and basically do similar things. So again, it's in a lot of research going on in this field, so it's very cool. We'll see how this.
With plasma that's like donated, does it matter about like the person's
Yeah, because let me think of how they do that. I don't know exactly what they do. They probably remove all the immunoglobulins if they're doing like plasma donations and you're getting plasma from someone, because you have to remove all those immunoglobulins, otherwise you're going to have a reaction. I don't know what that process looks like, but
what does it provide? Like
it's just tons of growth factors in plasma. Yeah. So usually when you do that type of regenerative procedure, you're going to use your own plasma, right? And so you can literally do this at even med spas. There was a place that I worked at where they would use like PRP to do like hair regeneration. So they'll like inject it into the patient's scalp, or they'll, inject it into like skin. Yeah, basically. I mean, you have all these growth factors in there that can really help to stimulate collagen formation with like fibroblasts and things like that. So super cool stuff. And similar techniques that could be used in neural regeneration. So here are a handful of growth factors and whatnot that can help to allow for growth of cells. So we're going to talk about this one in particular in a little bit. So the SOX transcription factor. There's also nestin, and let's get into that in a second. So we'll talk about that a little bit. So here's another image from your textbook. So they're injecting fluorescently labeled stem cells into an embryo, and they're being able to see how those stem cells could thereby get integrated into the central nervous system of the embryo. So you can actually see the fluorescently marked and dyed stem cells like glowing. So super, super cool stuff. All right, now these are the transcription factors that I want you to be familiar with. So these are the major four. So you have the SOX2 transcription factor, OCT4, KIF4, and CMYC. Those are the four major transcription factors I want you to be familiar with. And those are the ones that are able to actually be used to induce pluripotency into different types of cells. And we're going to be focusing here on fibroblasts, but to my understanding, there's other cells that you can also use that you can induce pluripotency into, but fibroblasts are going to be one of the most effective types of cells to do that. And there's other techniques too that are coming out that are a little bit more novel. This technique has been around for a little bit, I think like 15 or 20 years or so, but there's other stuff that's coming out of the woodwork nowadays, which is kind of neat. So let's watch a little video on this.
Over 50 years ago, it was hypothesized that terminally differentiated cells such as skin fibroblasts could be forced to take on a pluripotent state similar to embryonic stem cells. The basis for this concept was the observation that all cell types, with a few minor exceptions, have the same genetic code, and that the only difference between cell types is how the code is read. This ability to change the cellular identity of a differentiated cell to a pluripotent state is termed cellular reprogramming. After half a decade of research, it was recently shown that the forced expression of 4 transcription factors can reprogram skin fibroblasts to become pluripotent. The impact of this finding is huge. Beyond the importance for basic biology, it also overcomes the major ethical objections to the use of human embryonic stem cells for transplant therapy. In order for reprogramming to happen, 4 genes that encode the transcription factors OCT4, SOX2, CMYC, and KLF4 need to be packaged and put into a lentivirus. tissue culture medium containing purified virus is then added to a cell culture dish containing a high density of fibroblasts. And within that dish, something amazing occurs. A small fraction of fibroblasts become infected with all 4 of the transcription factor carrying viruses.
So depending on what kind of viruses you use, it actually can become a little bit. There's certain cell lines that now like fall into different levels of safety. So are you guys familiar with like BLS safety? Like level 1, BLS 1 is like basic lab safety standards. BLS 1 is like just something that's not really necessarily going to affect you and cause any sort of disease. Like all the bacteria, for example, if you took microbiology here and you're doing like plating with bacteria, even the E. coli that we use is like BLS 1, right? It's basically safe. I mean, if you ingested it, you'd probably like get sick, but for the most part, they're pretty safe. Once you start getting into like viral, basically allowing viruses to place like the transcription factors or express the transcription factors into a cell, depending on what kind of viruses you use, that BLS category starts to change a little bit. You can get up to like BLS 2. There's a certain cell line, for example, we really wanted to use as part of our research. There were like LUMS cells, and that was BLS 2. And unfortunately, GCU doesn't have BLS 2 capabilities because they use viruses to basically transform those cells. So kind of interesting. Yep.
So in this case, with these 4 different transcription factors, are we just talking about how they affect fibroblasts and genes into similar properties?
Yeah, so they basically revert the fibroblasts back to their original state of like a pluripotent cell.
So it's like kind of like reverse transcription, but reverse.
Yeah, you can think of it that way. Yeah, it's kind of like reverse engineering, sort of. Okay. Yeah, so it lets them go back in time, basically, reverts their original cell line. Yeah, so they call that induced pluripotent stem cells, or iPSCs.
How does Sendai virus reprogram cells? Induced pluripotent stem cells, or iPSCs, are genetically reprogrammed somatic cells that exhibit pluripotent stem cell state similar to embryonic stem cells. The discovery in 2006 that human and mouse fibroblasts could be reprogrammed to generate iPSCs with qualities remarkably similar to embryonic stem cells has created a valuable new source of pluripotent cells. Fibroblasts, similar to other somatic cell types, do not express high levels of the transcription factors OCT4, SOX2, KLF4, and c-MYC under normal conditions. High levels of expression of these 4 genes will cause reprogramming of the fibroblasts and it will become pluripotent. There are multiple methods to generate iPSCs. Viruses, such as retroviral vectors, require integration of the viral genome into the host chromosome to express reprogramming genes. Normal gene transcription allows the inserted OCT4, SOX2, KLF4, and c-MYC transgenes to be expressed along with the host genes. Integration of viral DNA into the host genome disrupts the genome of the cells. This alteration can render the iPSCs and their derivatives less safe for clinical application and can compromise compound screens or disease pathway analysis. On the other hand, Sendai virus is a single-stranded negative sense RNA virus that replicates in cytoplasm. This means it does not integrate into the host's genome. Sendai virus replicates independent of cell cycle, unlike other approaches where the exogenous genes are expressed only as the cell divides. Using this strategy, Sendai virus produces very high copy numbers of the target gene. The Cytotune IPS Sendai reprogramming kit contains 4 Sendai virus-based reprogramming vectors, each expressing one of the 4 Yamanaka factors OCT4, SOX2, KLF4, and c-MYC.
All right, so any questions on any of that stuff? So just be familiar with these 4, okay? And I spelled this wrong, KIF, it's KLF. So it's not KIF4, it's KLF4.
So they each like work together?
Yeah, I mean, I guess you have to have every single 4 of these combined to be able to actually do an induced pluripotent stem cell. So I guess if you miss any of these ingredients, you're not going to get pluripotent stem cell. So this is what these different transcription factors do. I'm not going to test you on the exact mechanism of each of these, right? But just to give you an idea of what they do. So for example, the SOX2 transcription factor helps with self-renewal, pluripotency of the undifferentiated cells. OCT4 also self-renewal. KLF4. So this is from your textbook. Let me see if it's interchangeable, if it's KLF or KIF. KLF versus KIF. Let me just verify that. Because it comes from the name Kruppel-like factor, so it's KLF. So that is a typo from your guys' textbook. So it's actually KLF4. So go ahead and fix that on your guys' notes to KLF4. My bad. Anyways, it's going to be important for DNA repair, replication, and then c-Myc is actually a proto-oncogene. c-Myc is used for lots of other different genetic type research and genetic pathways as well. But it's going to be involved in the cell cycle. So progression, apoptosis, transformation. In other words, it can be involved in cancer, right? That's why it's a proto-oncogene, right? So think about the cell cycle. Whenever you have cancer, that's going to be something that affects the cell cycle. The cell basically is able to circumvent the entire cell cycle because there's a couple different stops within the cell cycle. And so a cancerous cell can basically just continually grow uncontrolled. And so c-Myc might be involved in something like that if you had like a mutation on c-Myc. So again, here's how you induce your pluripotent stem cells using those 4 major transcription factors. Yes?
So we don't have to know like every...
No, I'm not going to test you on each. Just know which of the transcription factors are going to be really important for inducing pluripotent stem cells. So again, OCT3, again, KLF instead of KIF4. That's hilarious. So the textbook made a huge, even in their diagrams.
Especially if it's in a diagram.
Yeah. So anyways, KLF4. So you can scratch that off. And c-Myc and OCT. Yeah, it's OCT4. Yeah. Yeah, so OCT4. So just remember OCT. So it'll be OCT4. That's funny. I love it. So again, they use these retroviruses to be able to either integrate the genome of the virus into the DNA, or there's other types of viruses you can use that don't necessarily have to be integrated to the DNA. What's up?
What's a retrovirus compared to a
Retrovirus has to inject, or not inject, they have to basically splice their DNA into the host genome. So HIV is a great example of retrovirus. HIV, in order for it to actually be able to reproduce itself, it has to be integrated into the host genome. And that's why when you have HIV, that's it. Like, you have HIV for life. You're not going to... Yeah, basically. Yeah. And so that's kind of how a retrovirus works. Yeah, so there's like... and then for HIV, there's actually a lot of ways that you can target the virus, different steps of the infection. So integrase inhibitor is one of the types of drugs that you can use for HIV treatment. It prevents the integration of the HIV DNA into the host DNA. So that's one type of drug that you can actually use. There's a bunch of other drugs too, like protease inhibitors that prevent the assembly of the virus and all that stuff. Cool. Anyways, moving on. So what's an organoid? An organoid is basically a simple tissue engineered cell based in the vitro models. And it's just a simple kind of like a simulacrum of the actual like more complex type of tissue. And so when we talk about organoids in, for example, like Petri dish, right, you're creating a type of tissue that belongs to like, you know, a more complex system, right? But you're doing it more like in an engineered type of fashion. You're actually creating it in a lab within a Petri dish. And so induced pluripotent cells in a Petri dish, you can basically add transcription factor to the cells and you can create almost any type of cell line, right? And you can create organoids, which are going to be like more simple version of an organ system. There are other types of things that might affect the ability for induced pluripotent cells to be able to turn into organoids. And these are the different types of things that you can add to a petri dish to be able to grow those cells. So you can use fibroblast growth factor, right? So FGF is one thing that you can use. You're going to have different types of media. I'm not going to test you on this stuff, by the way, but this is how you would do it if you were like working in the lab. So we use, like, for example, we use the cells that we get. We actually purchase our own cells. We add growth factors to them. We add media to them, and then you can differentiate them with using things like retinoic acid. So retinoic acid would help to differentiate the cell line into a more specific type of cell line. And so this is what fibroblast growth factor does. Basically allows for proliferation, differentiation, and patterning of the cells. Media, that's just basically what the cell grows in. It's just a substrate, right? It just provides nutrients, for example, different types of nutrients that are necessary for the cells to actually grow and survive. And then, of course, your iPSCs, or human pluripotent stem cells. And then retinoic acid, huge in differentiating cells. So retinoic acid, where does it come from? It's vitamin A, basically. Vitamin A, lipid-soluble vitamin, right? And it's going to be really important for cell growth, differentiation, and as well as activating nuclear receptors. So here are a couple of examples, right? So here's an example of your retinal cells in the eyes, right? You got retinal ganglion cells. And there are, in this specific example, they have different markers that show you which different layers are which. So the blue ones are going to be your photoreceptors up here. Red, those are going to be your bipolar cells. Green, that's retinal ganglion cells. And so they were able to put in like markers to be able to identify each of these different cell layers. And then down over here in D, that's going to be part of the forebrain. And you can see all the different types of cells that basically develop the brain. So you have cortical progenitor cells, right? Progenitor just means any cell that can turn into something else, right? And cortical neurons in green. All right, neural stem cells. So when you actually differentiate a neural, one of those pluripotent cells into a neural stem cell line, these are the types of cells that they can create. Neurons, astrocytes, oligodendrocytes. Those are the main neuronal type of cells that can be produced. And again, here are some other transcription factors that can be also used. For example, BHLH, so that's going to be the basic helix loop helix transcription factor, and SOX2. Those can be used to basically induce specialization in those neuronal type stem cells. And here are a couple other images from your textbook showing things that you can do in a lab, right? In vitro type conditions. And so you can do cell culturing, for example. You can see over here in B, that's going to be on a little petri dish doing cell culturing. You can see all the neurites, the dendrites being formed. They can make synapses with each other. That's super cool. The cells actually that we're growing right now, we just added retinoic acid to them last week. Before that, they were just kind of like hanging out. They look a lot like fibroblast type cells. But once we added the retinoic acid, it started to do this. It actually starts forming neurites, and the cells now start to form synaptic connections with one another. So super, super cool. And then astrocytes, oligodendrocytes, those are some of your more mature, fully grown cells. These are going to be referred to as post-mitotic. So neurons, once they become mature, they're not going to be undergoing mitosis. So these are post-mitotic cells. They've already been differentiated at that point, right? They're not going to change any further beyond that. Oligodendrocytes as well. All right, now let's switch gears and let's talk about embryogenesis. So embryogenesis, the term that we're going to be using is gastrulation. And when you have gastrulation, that's when you're basically forming the three major layers that are going to turn into all different types of tissues as an individual develops and matures. Right. So again, the ectoderm. in terms of neuroscience or neurological process, ectoderm is what turns into basically the entire nervous system. So it's going to be forming neurons, it's going to be forming the brain, spinal cord, etc. So in other words, central nervous system is derived from the ectoderm, specifically the neuroectoderm. Then you have the mesoderm. That's going to be the middle layer. The mesoderm is where you get the notochord formation. And then the endoderm is where you get some other, enteric type organ systems being derived from that. So the endoderm is not going to be super important in terms of gastrulation for the neuronal system. Only the ectoderm and the mesoderm. Again, neuroectoderm is what forms the entire nervous system, and it's going to be derived from the ectoderm. And then when you have neurulation, neurulation is where you have a notochord being formed. And the notochord is going to be basically an invagination of the mesoderm. So that's where the notochord comes from, mesoderm. And that's going to be coming from the primitive streak. The primitive streak is basically what kind of bifurcates the developing embryo down the middle. And so the primitive streak eventually invaginates, and then that's where you get the formation of the notochord. So let's watch a little video on the process of gastrulation.
Shortly after fertilization, the fertilized egg or zygote begins dividing. By about 4 days after fertilization, a solid ball of cells called a morula forms. The cells continue to multiply, and by day 5, the cells separate into 2 groups, the inner cell mass, which will become the embryo, and an encompassing sac called the trophoblast, which will develop into the placenta. Trophoblast cells secrete fluid, creating a cavity, the blastocele, with the inner cell mass at one end. At this stage, the mammalian embryo is called a blastocyst. The cells of the inner cell mass eventually form 2 layers, the epiblast and the hypoblast. The epiblast is thought to contain the cells that will generate the actual embryo. Critical to the early development of all vertebrate embryos is the process of gastrulation, which occurs between days 13 and 19. Gastrulation results from the movement of cells toward the midline and then forward along the midline, forming a groove called the primitive streak. The end result is the production of the 3 primitive cell layers, the endoderm, the mesoderm, and the ectoderm. The nervous system develops from the outer layer, called the ectoderm. As this cell layer thickens, it becomes a distinct epithelium called the neural plate. Uneven rates of cell division form a groove called the neural groove that will become the midline of the embryo. Ridges of ectoderm continue to bulge on both sides of the midline. The tops of the neural ridges eventually come together, forming the neural tube. The interior of the neural tube becomes the fluid-filled cerebral ventricles of the brain, the central canal of the spinal cord, and the passages that connect them.
All right, cool. And then eventually, this notochord is what turns into the intervertebral discs within the spinal column. It can also turn into some other portions of the bones as well, a little bit of the nucleus pulposus, which is going to be within the intervertebral disc. But that's kind of what the notochord turns into. Basically supporting structures for the spinal column, effectively. There are some animals that literally don't have a spinal column, some fish, for example, that literally just have a notochord going right through them. One good example would be lamprey. You guys know, have you seen lamprey? One of the most ugly things on earth. It's like a worm that basically has these disgusting teeth at the end of it, and they latch on to fish as a parasite. They suck blood. They're called vampire fish as well. Ugh. Super ugly thing. But if you dissect a lamprey, you're not going to find a spinal column. You're going to find only a notochord, which is a cartilaginous structure that basically goes throughout the entire length of the animal. So this whole entire formation of embryogenesis, the process of embryogenesis is very, very similar across species, right? They have very similar processes, right? And a lot of the genes that we've studied, we're going to talk about that later on, like the Hox genes, the homeobox genes that basically allow for like segmentation of Drosophila flies, right? A lot of those same exact Hox genes are found in our genome as well that help basically to allow for segmentation, differentiation of tissue types. So super cool stuff. So when you study embryogenesis, there's people that study this across species, right? They do like comparative zoology, and they'll see the differences between different animals and how they develop. So it's really, really neat to see. So definitely make sure you are familiar with everything going on in this slide over here, because this is the kind of thing that I'm going to definitely test you on. So know which structures are which, especially the ones that I highlight over here. So you have your neuroectoderm, which effectively turns into basically all the cells within the nervous system. Right? Those are the ones that are going to eventually mature into the brain, the central nervous system, and all that stuff. The neural crest cells are the ones that basically start to form the tube. So once the neural crest cells migrate and you get that complete invagination, you're going to get them to meet, and then that eventually turns into the neural tube, which later turns into the spinal cord. And then another thing that's going to be important as well is you have your spinal cord as it develops over here, and then you have the anterior neural fold, and the anterior neural fold is what eventually turns into the brain. Right? Then you get all the different subsections of the brain when we're talking about the diencephalon, the mesencephalon, all that stuff. That's all going to be derived eventually from the anterior neural fold. And then another thing that I want you guys to be familiar with, and I didn't highlight it here, but you can highlight it on your slides, the neural crest cells. These neural crest cells eventually start to migrate down as the neural tube is developing, and the neural crest cells eventually turn into the peripheral nervous system. Peripheral nervous system as well as like sensory nervous system. So you got your afferents derived from the neural crest cells. So these eventually start to migrate throughout the rest of the body, right, turning into the peripheral nervous system. No, that's going to be peripheral nervous system specifically, and especially sensory, not central. Yeah, it's going to be your sensory afferent type of nerve fibers, and those are derived from your neural crest cells. So let's go through all of these again. So during late gastrulation, early neurulation. So early neurulation, that's where you start forming late gastrulation means you're starting to get some like more distinction within each of these layers, and that's when you start getting the neuroectoderm being formed, right? The neuroectoderm eventually turns into all of the nerves of the central nervous system. And then you have the notochord, again, that is from the mesoderm. Notochord from the mesoderm, eventually it turns into some important structures within the spinal column, right? I'm talking intervertebral discs, things like that. Then you have your neural plate, and the neural plate is a part of the ectoderm, and that's what basically turns into the neural ectoderm. Then the neural plate starts to fold. It starts to invaginate in on itself. And when it starts doing that, you start getting the formation of a neural groove, and then eventually the neural tube forms out of that. The other thing that you might want to note here is the floor plate. The floor plate is going to be the structure that's directly above the notochord. And the floor plate will eventually give rise to some of the more like, like somatic type cells, efferent type cells, motor type neurons. And then again, neural crest cells, these are the ones that eventually give rise to your peripheral nerve, your autonomic nervous system, peripheral nervous system, sensory, etc. All right, and then edges of the neural plate, they are going to eventually merge, right? So you get that invagination, and eventually it merges together, and then they meet in the middle, and then you have the neural tube finally being formed out of that. And then the neural tube, that's going to basically turn into the spinal cord. And then again, neural crest cells, sensory, autonomic system, peripheral nervous system, etc. And then again, the anterior end of the neural plate, that's what eventually turns into the anterior neural fold, which eventually gives rise to the brain in the mature individual. So here's a couple images that basically kind of give you an idea of what that progression looks like. These are all color-coded, so you can see different regions of these cells that turn even into like part of the bony structures in the face and the head as well. Here's another image that basically shows you all the different cell lines and derivatives and what they eventually turn into. So I'm not going to test you on anything like this. I just put this in here just so you can get an idea of what the progression looks like and what they eventually turn into in the mature individual. And then here's another schematic that basically describes the ectoderm and mesoderm components, right? The ectoderm, the neuroectoderm, you get the neural plate formation. Eventually the neural plate invaginates a little bit. Then you get the neural groove. And then the neural fold meets over. So it's like you're basically folding a burrito, right? And so you're getting both of those sides to connect. And then once they do connect at the midline, then you get the formation of the neural tube, which eventually turns into the spinal cord. And then all those neural crest cells migrate and turn into your peripheral nervous system, autonomic ganglia, things like that. Here's another image of that. So here's your blastula. And then the blastula eventually turns into the gastrula, and that's when you get the actual 3 different layers: ectoderm, endoderm, mesoderm, etc. And then neurulations, that whole entire formation of the neural groove, neural crest cells, neural tube, etc. So just another reiteration of all that. All right, now the neural stem cells the neural crest cells are going to be subdivided into 4 major groups. So you have the cranial contribution, vagal contribution, trunk, and sacral. Within the cranial, from the neural crest cells, the cranial cells are going to eventually turn into the bony features of the face and skull. Right, you get the cranial facial skeleton being formed from the cranial cells. You also get cranial ganglia, teeth, things like that are going to be formed from these types of cells. So if you go back over here, you can see all the bony formations. These are all going to be derived from neural crest cells. Okay, so neural crest cells, they do a lot of things. It's not just, peripheral nervous system, autonomic ganglia, and things like that. They also can help to eventually form some of the bony structures in the head. What else we got here? We have the vagal derivatives and contributions, and that's going to make up enteric ganglia. What does that mean? What do you mean when we talk about the enteric system? Yeah, your GI tract, exactly. Right. So these are going to provide the autonomic innervation that innervate the enteric system. Right. So think about the vagus nerve, for example. What does the vagus nerve do? It allows for like peristalsis to take place. Right. Whenever you swallow something, when your esophagus contracts, that's all peristalsis. That's going to be mediated by the vagus nerve. Right. So it's going to help mediate your GI tract and provide innervation as well to smooth muscles as well as the heart. So vagus nerve, think about the vagus nerve when you think the vagal derivatives. And then the trunk. And in the trunk, you're going to have dorsal root ganglia. So that's going to be your part of your sensory input into your spinal cord. You get your sympathetic ganglia. And then you have your adrenal medulla, which releases what? Yeah, exactly. Your catecholamines, epinephrine, norepinephrine. And then you have your sacral contributions. You also have some enteric ganglia as well. I would imagine that would be probably the distal end of the GI tract, so maybe the colon, large intestine. And then you also have some of the sympathetic ganglia derived from the sacral contribution. So know these 4 major contributions of the neural crest cells, so cranial, vagal, trunk, and sacral. Those are going to be really important. All right, so here's a couple other images to basically demonstrate neural crest cells and what they look like in a developing embryo. So all this labeled here blue, those are all going to be derivatives of the neural crest cells and how they migrate to eventually turn into these different subsections, right? And then you have sensory structures, you have sensory ganglia, right? Those are going to be all derived from your neural crest cells. And they listed some of the important transcription factors. So MYC or MYC is one, SOX, right? We already talked about 2 of those different types of transcription factors when we're inducing pluripotent cells, right? So these are going to be really important transcription factors to allow for the differentiation of the neural crest cells. So again, here are your 4 major contributions: cranial, vagal, trunk, and sacral. And then here's the migration of the cells. So you have the cranial migration, right? That's what allows for all these different cells to eventually turn into like the bones in the face, for example. And then you have your trunk migration that's going a little bit further down to form, for example, the sympathetic ganglia. So cool. Let's move on. Here's some other things, other structures that are going to be some of the that are derived from the neural crest cells. So you have your nose, eyes, pinna of the ear, all that. Okay, let's move on. These are just some images from your textbook to further illustrate that. All right, now let's talk about your embryonic brain derivatives and what they eventually turn into as the embryo develops and then eventually into a mature adult, for example. By the third trimester, that's when your spinal cord and brain are basically fully, clearly demarcated, fully separate, right? They actually have their own unique structures, right? It's not just a big blob of cells at that point. Now it's actually like fully developed. So that is during the third trimester. And the main derivatives are going to be these three, right? You got the prosencephalon, the mesencephalon, and the rhombencephalon. For the prosencephalon, that's what eventually turns into telencephalon and the diencephalon. The telencephalon, if you guys remember, that's what makes up like your cerebral hemispheres, for example, right? That's going to be your like forebrain, your frontal lobe, right? Your parietal lobe, all that. That's going to be derived from the telencephalon. Diencephalon, again, thalamus, hypothalamus, epithalamus. And then mesencephalon is going to be your midbrain, so that's completely separate from these. But then you have your rhombencephalon, and the rhombencephalon is what makes up the metencephalon and the myelencephalon. What does the metencephalon turn into when it matures? Yeah, cerebellum is one. What's another? Yes, and the pons, exactly. So that turns into the pons and cerebellum. What does the myelencephalon turn into? Yeah, exactly. So it's going to be the medulla. The medulla derives from the myelencephalon. So all those different structures are already clearly demarcated by the third trimester. Yes?
Wait. But I was going to ask what the telencephalon turns into because I missed what you said.
Oh, yeah. Telencephalon it basically turns into all the higher functioning structures, right? The cerebral cortex, also the cerebral nuclei, right? That involves like the basal ganglia, so you can control things like movement, amygdala, which is part of your limbic system, all that stuff. So telencephalon, super important structure. But that all falls under the category of the prosencephalon. So here are the structures as they develop in embryogenesis. So at the onset, in early gestation, you're going to have this kind of like a cane-shaped structure, right? Where you're going to have the prosencephalon, which eventually turns into the forebrain, and then you have the mesencephalon connecting to the rhombencephalon, and all the way down to the spinal cord. And so make sure you're familiar with what these different color-coded areas represent and what they eventually turn into. Either this image of embryogenesis and gestation, or this kind of like more, what would you call like an idealized diagram of those different regions. And then you have the spinal cord. Again, that's going to be originated from the neural tube. So the neural tube eventually turns into the spinal cord. Then the next thing that you see is that the prosencephalon eventually basically turns into the telencephalon and diencephalon. Okay, so the origins are the prosencephalon. And then the rhombencephalon divisions, again, they turn into the metencephalon and myelencephalon. Metencephalon, pons, and cerebellum. Myelencephalon, the medulla oblongata. And then you also get ventricles being formed in these regions as well, right? We already talked about all those different ventricles: lateral ventricle, third ventricle, fourth ventricle, etc. Where are we at with time? And then let's see here. Fetal brain and spinal cord, again, at the end of the second trimester, basically the third trimester is when all these things are going to be definitively developed, right? So let's go ahead and do some quizzes questions, and then we will continue on if we have time. So any questions before we do some questions? All right, cool. Let's do it. Let's go ahead and log in. Anyone having trouble logging in? Yeah? Is your internet slow or something?
Yeah.
I'm going to go ahead and hit start. You can still log in even after I start it, so let's go ahead and begin. All right, softball question. Ultimate precursor for generating tissue-specific stem cells? Yes, pluripotent stem cells. Perfect. All right, stem cells are generated by the first series of cell divisions of the fertilized oocyte. So those are going to be your pluripotent stem cells. So let me go back to the slides. Okay, let's go back to that image that I had at the very beginning, right there. Okay, so pluripotent, that's going to be what you have after the actual blastocyst is being formed. Okay. Oh, did I word it incorrectly? Because totipotent is going to be before, right? Yeah, it's totipotent as the cells are starting to divide, right? You have the morula, which kind of looks like a mulberry, right? You got a morula formation, and then eventually it turns into the blastocyst. The cells within the blastocyst, that's after you get that division, that's when they turn into pluripotent cells. Let me see how I worded it. I might have worded it incorrectly. Let me just cells that do first cell division of fertilized eggs.
I think that's just
Yeah, I can see how that was confusing. Let me look. I'll go back on that question, see if I can reword that so it's a little bit more clear.
Are you going to be testing on the intermediates between totipotent to like pluripotent and
Maybe. I'm still writing the exam. I might put that in there. Just give me 1 second. Sorry about that. Okay. All right, so I can remember to fix that later since, yeah, let me fix the wording on that. right, cool. Another softball question, right? Using IPSCs, those are the ways that you can really be able to avoid any sort of issues when it comes to like ethical dilemmas. So good job on that. I know, I'm going to fix that. So KLF4. Good. Yeah, no one clicked HOX, which is a gene, right? That's going to be part of the homeobox genes, which we'll talk about in a little bit. All right, cool. Yep, fibroblasts. That's going to be one of the major cell types that you can induce. What are myeloblasts? You guys familiar with like AML or CML? Or you should be. We just talked about it in your pathology class. Huh? Your test is like in a couple days, so you better remember. So myeloblasts, those are going to be the progenitor cells that basically turn into like red blood cells as well as your granulocytes, right? So like so if you have like AML, which is acute myeloblastic leukemia, so these are basically immature cells that would have otherwise turned into like granulocytes. So myeloblasts would not be one of these cells that you would induce. Fibroblasts would be the best pick. Sorry, I didn't mean to single you out. All right, cool. Absolutely. Astrocytes, oligodendrocytes, neurons, right? Not myeloid or lymphoid cells. Those are going to be more of those types of cells that would turn into leukocytes, white blood cells. All right. Softball question. 3 layers: ectoderm, mesoderm, endoderm. Let's see if that was a softball question. Good. Most of you guys got that correct. So yeah, that was the notochord. So F. It's going to be the notochord. I got another question about that. Next. All right. Good. 2 of you guys got that correct. That's perfect. Mesoderm, okay? Notochord, that makes up, again, parts of your spinal column, right? For example, your intervertebral discs, right, that goes between each of your vertebrae. That's going to be derived from the notochord. Okay, so it's not going to be forming really like the neuronal tissue itself, right? It's not going to form the central nervous system. So it's not going to come from the ectoderm. It's going to come from the mesoderm. So good. Well, good for the two of you who got that correct. So for the rest of you, make sure you know that the ectoderm is what makes up the rest of the neuronal development, right? There's actual brain tissue and things like that, neurons. Cool. Let's see. Structure labeled as B. Okay. Basically, everything else is going to be ectoderm for the sake of neurogenesis, right? So ectoderm turns into neuroectoderm, which turns into basically everything that forms the brain, spinal cord, etc. So if it's not the ectoderm, then it's probably the mesoderm, in other words, right? Notochord, mesoderm, everything else, ectoderm. All right, good. Let's see. With cells listed as A are referred to as what? Good. Most of you guys got that correct. So those are your neural crest cells. Okay. All right, let's go to the next one. Another question here on neural crest cells. Cool. Neural crest cells eventually transform into which structure? Select all that apply. Not the central nervous system. Okay, neural crest cells do not form the central nervous system. So the central nervous system is going to involve like the neural tube. It's going to involve the anterior fold, right? It eventually turns into like the structures of the brain. So it doesn't form the central nervous system. It doesn't form the motor neurons. The motor neurons are going to be derived basically from all the other cells, like the neural tube, etc. Right? Neural crest cells will form the peripheral nervous system, and they're going to form sensory neurons. So the sensory ganglia, for example, that's going to be derived from neural crest cells. So make sure you know those derivatives. All right, good. That was pretty easy, right? Cranial, vagal, trunk, and sacral are going to be the major neural crest contributions. A couple more questions. Another softball question, right? Third trimester. I hope someone said fourth trimester. Dang it. All right, let's do a couple more. All right, let's take a look here at the image. So the structure listed as A is the blank and eventually gives rise to what structures? So A, what is that structure? Yes, prosencephalon. Good. Prosencephalon. And which is the correct answer here? Forebrain. Right. The prosencephalon eventually turns into the what? What are the two major things that it eventually turns into?
Cerebral cortex.
Yes. What's the fancy?
Telencephalon?
Yes, telencephalon. Yeah, then you get the diencephalon as well. So that's what the prosencephalon basically contributes to eventually. All right, last question. All right. It's quite a spread. The structure labeled as C is referred to as the blank and eventually gives rise to what structure? So C, what is that structure called? Yeah, that's the rhombencephalon. Good. That's the rhombencephalon. And the rhombencephalon eventually turns into what? The what? Myelencephalon and metencephalon. Yeah. And then you have the mesencephalon, which eventually turns into midbrain. Good. Excellent. So make sure you're familiar with those different structures. If I have pictures like this, probably going to have quite a few pictures like this on the exam. So it's going to have different labels, different areas in embryogenesis, and know which what they eventually turn into in the adult brain. Any questions on any of that? Cool. All right, let's continue. We got 15 more minutes. All right. Now, this is where the fun really begins, huh? We can start talking about genes. I'm not really going to test you on like all the genes though. Like you just need to know some like basic overarching concepts, right? So here's one thing, neuromeres. Those are going to be basically distinct units of the nervous system. And it's going to have specific anatomical contents associated with each. And the way that neuromeres are subdivided is going to be through those homeobox genes, also referred to as Hox genes. And there's a bunch of Hox genes. You're not going to be responsible for all of them. But basically, Hox genes allow for subdivision as development takes place. And these Hox genes, again, these were discovered in Drosophila invertebrates, right? And they're going to have segmented bodies. And these Hox genes basically determine the segmentation as the fly develops, right? And we have a lot of those same exact Hox genes in our genome as well, and effectively does similar type of things, right? It helps to, develop certain regions of your brain, nervous system, and the rest of your body as well. So these sequential gene expression that's going to be associated with Hox genes, again, also referred to as homeobox genes. And I'm not going to test you on these genes over here, but this basically shows you the sequence of genetic expression as you have the larva turning into an adult. So this is all based off of the development of a Drosophila, which is where we get a lot of our information from in terms of genetics, right? Drosophila, C. elegans, lots of really cool little tiny creatures that have similar genes as we do. So pretty cool. And then the benefit of using Drosophila. What's the benefit of using Drosophila? Why do we like to use animals like this? Yeah, they reproduce really, really fast, and you can easily put in knockout genes, knock-in genes, right? You can basically toy with their genome, and you can see what the results are. So that's one of the reasons why Drosophila have been so popular in genetics. So anyways, this is a series. So there's bicoid, so that's what allows for anterior pole differentiation. You have the Krupel gene, which allows for anterior-posterior axis, hairy gene, wingless gene. Some of these genes are also named after, like, when they wiped out the gene, what did it do? Maybe the wingless gene resulted in 0 wings on the Drosophila that were born, right, without the wingless gene, or WG. But that also allows for organization of the segmentation. And here's your Hox genes. So the Drosophila karyotype, I believe it's 8 chromosomes, if I remember correctly, but not very many chromosomes compared to humans, right? We have quite a few more, right? 23. With our chromosomes, you can see exactly where the Hox genes correspond with the Drosophila's Hox genes. So you have your ancestral Hox clusters, and then if you look at ours, our chromosomes, it's a little bit more, spread out. Like, you have some that are going to be seen on, like, chromosome 7 or chromosome 2 or chromosome 12. So they're going to be spread out amongst the different chromosomes, but effectively they do the same thing, and they correspond with the Hox genes that you find in other species as well. So super cool that you see all that crossover, right? These are like highly conserved regions of genetics, right? If you were to even compare the genes of a human to the genes of a rat or a mouse or a rodent, right? I forget what I think it's like 96 or 97% conservation of genes. Like, we have a lot of the same exact genes, right? There's only minor variations. Right? Those minor variations, of course, make a huge difference, especially when we're talking about diseases. We're going to talk about that later on. So there's going to be a couple genes that I want you to be familiar with when it comes to, like, clinical application. All right. So rhombomeres, right? Rhombomeres are going to be these little sections, right? They're transverse sections, and they go along longitudinally, right? So they're going to be longitudinal sections along the axis, and they have these little bulge appearances, right? And so this is what basically allows for the brain to start becoming segmented and develop, right? So, and these are all going to be based off of Hox gene expression, right? So the Hox genes basically allow for different definitive boundaries within the different regions of the brain as it develops. And then you have the hindbrain developing, and that eventually will allow for differentiation of cranial nerves, right? Remember, cranial nerves are going to be coming off of your brain stem, right? So that's part of your hindbrain. And then you also have different ganglia that are going to be associated with that. So like dorsal root ganglia, for example, as you go further down towards the spinal cord. So here's your hindbrain. So on the left over here, these are all your sensory ganglia and nerves. These are all going to be the motor and mixed nerves. So they were able to separate those two out so you can see what's going on there. So you have the trigeminal ganglia, which eventually gives rise to the trigeminal nerve. What else? Let's see here. You got your cranial nerve 10. The ganglia are going to be coming off there. Jugular, dosa ganglia. Again, I'm not going to make you remember the terms of all these different ganglia, right? Just make sure you know the cranial nerves and what they do. But then you have the motor ones, like facial motor nerve, right? Facial nerve is what allows for you to have expression in your face, part of your cranial nerve 7. Abducens, which is part of your cranial nerve 6, allowing for you to abduct your eyes. So these are all going to be basically demarcated by Hox genes, right? Hox genes are going to be important for all that. What else is important here? Yeah, that's basically it. Let's move on. So you're going to have organizers. So these are going to be a type of, these are going to be things that allow for the organization of the different brain structures. So organizers are what induce the nervous system from an undifferentiated blastula. And they're going to be regions of cells, right, in an embryo that get induced, right? So when you induce, you're going to be effectively inducing the fate of the cell, what it eventually matures into, right, before it becomes post-mitotic, right, when the cell can stop dividing and can stop growing. Then you're going to also affect the patterning, right? So it's basically allowing for like organization, structural development, things like that. So those are going to be really important for organizers. So it basically organizes the nervous system. And then you have your germ cells, cells in the adjacent germ layers. And then you have your neural induction types of genes as well that can induce your different types of cells. And so here's an example of an organizer and how you can modify or you can like toy with organizers. That's what they did over here with these poor little tadpoles. So here's like a normal tadpole over here. And over here, they're basically messing around with the organizers. So you have different regions of the developing embryo that are going to be associated with different parts of the body. So you have the ventral division versus the dorsal division. And then that allows for, like, you know, basically dictating the migration of cells, right? Movement of cells, organization of cells. Now, here is an example of where they basically messed with the organization of the cells. So over here, they took off the ventral division and they added another dorsal division. And so you had two different dorsal divisions for this embryo. And then all of a sudden, you have two individual fish now fused within their ventral part of the body. So this was where they dissected a part of the embryo that was allowing for migration of the dorsal division. And then you have a second notochord basically being generated from that. So that's kind of an example of how you can mess around with the organization of cells, organization and the migration patterns of cells. So here's your normal and here's your genetically modified tadpole. And so here are some of those ways that you can induce those signals. So we already talked about retinoic acid. Retinoic acid, super important for allowing for differentiation of cells, right? Organization of cells. And then you also have your fibroblast growth factors, right? That's also going to be really important for patterning. So you have patterning of your axes during embryogenesis. And then you have bone morphogenic protein. Now, this one is going to be associated with chordinin and noggin. Noggin is a great name, right? So these are both going to basically regulate your bone morphogenic protein, and they basically act as antagonists. And so NOG, that's going to be a gene that basically affects the ability for bone morphogenic protein to operate. And then you have noggin, effectively is what controls that whole entire process taking place. And here are some other really important nerve induction signals. Sonic Hedgehog, just like the video game, right? Sonic Hedgehog is going to be really important for a lot of things. First, it helps to allow for patterning of the CNS, but it also does help in migration patterns of cells. If you affect the Sonic Hedgehog signal or SHH, that can cause all sorts of different diseases, which we're going to talk about later. We're going to discuss, for example, like prosencephaly, which can be holoprosencephaly, which can be affected by mutations of the SHH gene or the Sonic Hedgehog gene. Beyond the nervous system, Sonic Hedgehog is also involved in like the migration patterns of like organs, the different cell types that turn into different organs in your thorax as well as in your abdomen. So it's a very important gene for migration. Then you have the WNT gene. That's again also important in migration of cells, patterning, and all that. So you notice there's a lot of crossover in a lot of these different types of transcription factors. So super important.
These are all like antagonists. So they like
No. The only thing that's really the antagonist here was chordin and noggin. They antagonize the bone morphogenic protein. But these other ones are going to be basically chemical signals that are like, they serve sort of as transcription factors. So it allows for movement of cells, differentiation of cells, right? Specialization of cells, all that stuff. So you have to have all these transcription factors working in concert to do various things. So here's another schematic from your textbook that talks about all the different transcription factors involved in the roof plate, right? And you also have the floor plate. So the roof plate and the floor plate. The floor plate is what's going to eventually give rise to more of your, your motor type neurons, right? So you have motor neurons coming from your floor plate versus the roof plate is going to be more associated with like dorsal column, right? So you can think of this as an individual like laying, kind of facing down, right? So if they were like pronated or something. So this would be basically their back, right? And so these eventually are going to turn into dorsal column. This is eventually going to turn into the ventral part of the spinal cord, for example. So that's going to eventually give rise to your motor neurons. And so these are the different types of transcription factors involved. Look, notice retinoic acid is going to be commonly seen, affecting all these different migration patterns and specialization. I am not at all, you can even put an X through there if you want. I'm not going to test you on any of these different pathways involved in retinoic acid, fibroblast growth factor, and all that stuff. We are later on going to discuss, for example, tyrosine kinase. We're going to discuss tyrosine kinases when we get into the development of neuronal circuits, because tyrosine kinase is going to be really important in a lot of these different pathways. But I'm not going to test you on each individual pathway of these transcription factors. Just know that the transcription factors basically serve as ligands effectively, and then they bind to receptors and they allow for different signals to take place. And these signals do various things, right? Gene expression, things like that. Okay. Oh, you can just like scratch off this whole entire schematic. Like, I'm not going to test you on any of these pathways, right? That's like way too much granular information. So just be familiar with the transcription factors. You know, if you see retinoic acid, know that serves as a transcription factor. Right? Fibroblast growth factor is a transcription factor. And then these transcription factors help to establish neuronal identity. Okay, and that's what basically determines kind of what the neurons are eventually going to do once they're fully matured. That's kind of the whole idea of these transcription factors. Super important for movement of the cells, patterning of the cells, and the identity of the cells. And that's what we're going to close on today. We'll continue here next time on Thursday. And then we'll begin neuronal circuits. Cool? Any questions? All right.